Most of biochemistry is happening in an aqueous or watery environment, but we have those environments partitioned off by lipid membranes. So we have membranes surrounding our cells. We have membranes inside of our cells, making compartments like mitochondria, Golgi bodies, endoplasmic reticulum, all these various compartments. It's really important that we have that compartmentalization. But in order to have that compartmentalization, we've got to have these lipid barriers that prevent just the free flow of most molecules. However, there are molecules that we need to get inside and outside of cells. We need to get inside and outside of the mitochondria in various different other membrane-bound compartments inside of our cells. So how are we going to do this? How are we going to get those molecules that are often hydrophilic that want to be in that aqueous environment? How do we get them from one aqueous environment to the other aqueous environment if we have to go through one of those partitions, those membranes that are going to be those hydrophobic lipids? Not only that, but we have to let certain things through, but not other things. Otherwise, it wouldn't be very helpful compartmentalization, right? So how are we going to do this? Well, like most things in biochemistry, it depends. It depends on a few things. One is going to be the properties of the molecule. And so the chemical properties, how hydrophilic is it versus hydrophobic? How big is it? And the other is going to be whether we're moving along or against a gradient, or specifically the electrochemical gradient. And so we're going to get much, much more into this. But you can think about gradients being the molecular motivation to move. Molecules are going to move from high concentration to low concentration, as well as negative charges are going to move to positive charges, and positive charges are going to move to negative charges. We can compare both the concentration of molecules on either side of a membrane, as well as the electrical charge on either side of the membrane and the charge of the molecule, in order to come up with an electrochemical gradient that is going to dictate whether the molecule is going to want to go to one side or another side in terms of its thermodynamic favorability. So if a molecule is moving along its gradient, if it's going from high concentration to low concentration, from negative to positive, but we'll see it's like kind of a combination of the two of those things. If it's moving along its electrical chemical gradient, it's going to have a negative delta G that's moving downhill, basically. It's thermodynamically favorable. Its movement is actually going to produce energy. And we can say that it can move via what we call passive transport. So we'll see there are multiple types of passive transport, but what these have in common is a molecule is moving along its gradient. It's moving in that thermodynamically favorable direction. And it might not be able to get through the membrane by itself, but if we provide a channel, then we can get the molecules to go through just kind of on their own energetically. So we might have to provide the specific channel for them. We might need to open that channel, but then once that channel is open for them, then they can go and they can go through via diffusion. Conversely, what if we want to move a molecule against its gradient? When we're talking about moving it against its gradient, this is going to be energetically unfavorable. We're going to have a positive delta G. We're going to need energy. We're going to need to do what we call active transport. One way we can do this active transport is by coupling the movement along a gradient, which releases energy, to the movement of another thing against this gradient, which requires energy. That's what we call secondary active transport. And we'll also see that there can be primary active transport, where we talk about pumps that are typically spending or hydrolyzing ATP or another high energy molecule in order to transport a molecule against the gradient, typically by causing a conformational change in a pump that is then going to let that specific molecule through. So these are some of the key terms that we're going to be talking about, and we'll go much more into each of these. But this is the bottom line. We have passive transport and active transport. Passive transport, here the passage for you is free because you're thermodynamically wanted on the other side. You're moving along your electrochemical gradient. There are different flavors. So with simple diffusion, you just sneak in. Whereas with facilitated diffusion, a bodyguard. So typically this is in the form of a membrane spanning protein, some sort of channel helps you get through. These are often going to be ligand-gated or voltage-gated channels, so they're not just always open. Instead, they have to have a specific signal that tells them to open up so that you control the flow of the molecule. And they typically are going to be very selective to only let certain molecules through. On the other hand, we have active transport. Here, you're not thermodynamically wanted on the other side. You're moving against your electrochemical gradients, so the passage is going to be needed to pay for energetically. There's primary active transport, where you pay for your own entry or exit at the gate, often via the use of ATP or another NTP, and these are typically going to involve pumps. Secondary active transport, here your friend is paying ahead for you. So you have primary active transport of one molecule that's going to create a gradient to drive the co-transport of a second molecule. So you make a gradient of one molecule, and then you transport that one molecule that you pumped out back in, along with the molecule that is moving against its gradient. So we go down that hill in order to go up the hill for another. Quick note about how we talk about ATP. We're going to talk a lot about ATP, but it's really important to keep in mind that although we talk about kind of burning ATP or spending ATP, the breaking of ATP, the breaking of a bond is always going to be endergonic. It's going to always take energy to break a bond. But what turns out that the hydrolysis of ATP, the interaction of ATP with water to break off one of those phosphate groups, that's going to be highly energetically favorable. So although you have to put in a little bit of energy in order to actually break that bond, you are going to get much more favorable things in return. So remember, that for determining whether something is thermodynamically favorable, you need to compare the before and after. And so if we think about ATP plus water versus ADP plus this orthophosphate group, and all the interactions that we can have with this group, things like resonance stabilization, as well as things like freeing the repulsion of the charge between these groups. Therefore, we're going to have more stable products than reactants, and the kind of burning of ATP, the ATP hydrolysis, is going to be energetically favorable. So there's lots of nuance and stuff that goes into it, and we'll see later how keeping the ratio of ATP to ADP in our cells really out of whack so that we have a high concentration of ATP is going to make it so that this is even more thermodynamically favorable. We'll see that ATP is going to be a very powerful source of energy. We're going to store energy in the form of this ATP. And then we're going to be able to kind of spend that ATP. We're going to be able to hydrolyze it to cause a conformational change of our transporter. We're going to be able to phosphorylate a transporter to change its shape. We're going to be able to power all sorts of metabolic reactions using the energy that we've kind of stored in that ATP. So we're going to talk a lot about ATP, but just keep that in mind that it's not the breaking of the bond that is energetically favorable. It's what we get in return. And again, we'll talk a lot more about this. But by controlling the concentrations of ATP and ADP and all these various things, we can then also use ATP as a regulatory molecule in order to control when we're building up molecules, anabolism, versus breaking down molecules, catabolism. So lots of exciting things to look forward to in our metabolism section. A lot of metabolism, speaking of metabolism, happens in the mitochondria, which is one of the membrane-bound compartments. And so we need to start thinking about our membranes and our membrane transport. So remember that cell membranes aren't just lipids. So about half the mass of some of our membranes comes from proteins. We have a lot of proteins inside of those membranes, which is great because we're going to need those proteins in order to get many molecules through. So membrane proteins play important functions. They can serve as receptors, as signal regulators, as scaffolds, as cell-cell and cell surface adhesion molecules, and what we're going to be focusing on today as transporters, allowing molecules to move from one side of a membrane to another. This can be via active transport or via passive transport. Let's talk a little bit more about these proteins, and then we'll get into the different types of transport in a little more detail and look at some specific examples. When we talk about membrane proteins, that's actually a pretty broad class. We can think about things like integral proteins, integral membrane proteins, or IMPs, that go through a membrane. as well as proteins that are only associated with one side of a membrane. We can have proteins that are anchored on through a GPI. And so this is one of those examples of a glycolipid. So where we have this glycan, the sugar chain that's attached to a lipid, and then that's attached to a protein. So often we're going to have proteins that are anchored to a membrane via a glycosyl phosphatidylinositol or GPI linkage. And so that's actually not going through a membrane in that case. When we're talking about needing to get things through a membrane, we're going to want to have one of these integral membrane proteins, something that actually goes through the membrane so that our molecules can go through the membrane. But these peripheral proteins, these peripheral membrane proteins, these ones that are anchored or just hanging out on one side, are also going to serve important roles, just not be the focus of us right now. Speaking of these integral membrane proteins, these ones that go through a membrane, it's important to remember that the part that actually is facing the lipid is going to have to be hydrophobic. So that although we normally talk about proteins as having their hydrophobic parts on the inside and their hydrophilic parts on the outside, what's going to happen is that if you have one of these membrane spanning proteins, the part that is actually in contact with the lipid is going to have to be hydrophobic, but the parts that are in contact with aqueous things are going to have to be hydrophilic. Which parts are going to be dealing with aqueous things? Well, if we have a pore through it, then the center of the pore will need to be hydrophilic. And then the parts that stick out on either side of the membrane, so the part that's outside of the membrane, we call the ectodomain, and then the part that is inside of the membrane is called the endodomain. So like if this was a cellular membrane, this would be outside of the cell, the ectodomain, and inside of the cell, the endodomain. And then this would be the membrane spanning region. And we can have conformational changes happening in the ectodomain or in the endodomain that are going to facilitate movement through the channel that goes through the protein. Speaking of those channels, how are we going to make those channels out of protein? It's going to involve traversing that membrane through multiple passes or multiple times, basically, that you go back and forth through the membrane, often as alpha helices or beta barrels. It's important, though, to know that a single alpha helix, although it looks like there's a pore there, it's not. And so if we show this same alpha helix, so I've shown it in this cartoon mode from the side view as well as from a end-on view, and then I showed it in space filling in these spheres to actually show the size of the various atoms occupy. And you can see there's really not space for something to go through. So instead, we're going to have multiple alpha helices. You don't need to worry about knowing these actual numbers for this class, but for your alpha helices, you're typically going to have 7 or so of these alpha helices that are spanning the membrane in order to form one of these channels or pores. And each of these helices, each of these passes through the membrane is going to have 20 to 30 amino acids. And then you have your ectodomain on the outside, your endodomain on the inside, and these are going to be hydrophilic, but the parts that are actually facing the lipids are going to be hydrophobic. You can also have pores made up of beta barrels, and again, here it's where we have multiple beta barrels forming a pore, not a single beta barrel forming a pore. They typically have about 7 amino acids per pass because things are more spaced out in one of those beta strands than in an alpha helix. And at least every other one of those is going to be hydrophobic because you have that alternation of the side chains on one side of the beta strand, and then the other side of the beta strand, and then one side, and then the other side. So that was the reason why we could have those amino acids that were bulkier because they weren't going to be sticking right next to each other. And it's the reason why, if we have one side facing a lipid and one side facing an aqueous environment, every other one would be hydrophobic and versus hydrophilic. Now that we know a little bit about the structure of these proteins, let's talk more about how they go about their functions. Going back to our idea of separated gradients leading to potential energy, so you have a ball at the top of the hill, rolls down the bottom of the hill onto your toe, bangs into your toe, breaks your bone. Okay, that's a really bad analogy. But the energy from that movement was such that you were able to break that bone. And the energy from the movement of a molecule can be used to do much more happy things, like move something against its gradient, say. But you have this idea, this potential energy, this potential chemical energy that you can then use in order to drive unfavorable processes, such as the movement of a molecule against its gradients. So back to our gradients in a little more detail. Our chemical gradients, this is where we're going from high concentration of a molecule to low concentration of a molecule. This is just good old diffusion. So we have this net movement from high concentration to low concentration, but it's really just via this random movement. So random movement is going to lead to this even distribution via diffusion. That's going to be specific for a certain molecule. And so each molecule is going to have its own chemical gradient. But then you're going to have an electrical gradient that's shared by all members of the environment. That's coming from the sum of the charges of all of the molecules, of all the things that are going on inside and outside of the cell, or on one side of a membrane and versus the other. Inside of our cells is typically going to be more negatively charged than outside of our cells. So we say that our cells have a negative membrane potential. And when we talk about membrane potential, we're often going to be talking in terms of millivolts. We're not going to worry about numbers and calculations and things, just relative values. And so know that it's more negative inside of our cell typically, but what can happen is we can have, especially in our nervous system, we can have electrical impulses that change the polarity of the cells. So change the relative charges inside and outside of the cells. But that's our membrane potential. And then you can see that we also have concentrations that are different inside and outside of the cells. Let's take each of these into consideration because we need to think about an electrochemical gradient, not just an electric gradient, and not just a chemical gradient. Because as we'll see, sometimes they can actually be opposing. So if we take our sodium and our potassium, both of these are going to be positively charged, so they're going to want to go into the cell based on charge alone. So if you had equal concentrations of potassium, say, inside the cell and outside of the cell, there wouldn't be any chemical drive to move, so your chemical gradient wouldn't be contributing, but the electrical gradient would make it so that that positively charged potassium would want to go into the cell. But if we think about the concentrations that we typically find, well, here we're actually going to find that inside of our cell, we have a high concentration of potassium compared to outside of our cell. So outside of our cell, we've got about 4 millimolar compared to inside of our cell where we've got about 140 millimolar. And as we'll see, we have to use active transport in order to make it so that we keep that gradient. But we have a lot more of this potassium inside of our cells. We've got our special K. And so we have a unfavorable chemical gradient, but a favorable electrical gradient. And so we need to consider the electrochemical gradient to see kind of what wins out. In the case of our sodium, we have a higher concentration of sodium outside of the cells than inside of the cells, so it's kind of the reverse. So we've got 12 millimolar inside of our cells, 145 millimolar outside of our cells, and this is just kind of an average value, I'm assuming. So in this case, you have that the electric gradient is going to be driving things into the cell, and the chemical gradient is going to be driving things into the cell. In terms of our sodium, but in order to keep the sodium concentration higher out here, we're going to have to actually pump the sodium out. And we'll see how that we can do this. But for now, just know we've got chemical gradients and electrical gradients, and the combination of the two gives us an electrochemical gradient that is specific to a certain molecule in a specific electric environment. Those are both charged molecules, but what if we have an uncharged molecule? Well, we can still think of an electrochemical gradient, but the electric part of it is going to be zero. And so the equation for calculating the free energy change for transport of a molecule, so this delta GT, don't worry about all these numbers. Just know that this Z is going to be the charge. This is a constant, this Faraday constant, and this is the transmembrane electrical potential in volts. And so here we've got our electrical potential that's being multiplied by a constant as well as multiplied by the charge. And then here are your concentrations on one side versus another. You're typically always going to have concentrations on one side or the other, and then you're going to have this concentration part, the chemical gradient part, come into play. But if your charge is zero, this part is going to be zero, and you don't need to worry about that. So we can think about an electrochemical gradient for any molecule. And so we're going to be talking in terms of an electrochemical gradient. And it's this gradient that's going to power the movement of molecules, or this gradient that is going to impede the movement of molecules. If molecules were able to just freely move along their gradients, eventually there'd be no gradients. So we need our membranes to be semi-permeable. So in terms of semi-permeable, that means that a membrane will let through some things, but not other things. So cell membranes are only freely permeable to nonpolar molecules, small ones, so they can't be too big, as well as small polar but neutral molecules. It's kind of weird that water can actually go through the membrane freely. It also has channels called aquaporins to help more go in and out, but water is small enough that it can go through, even though it is kind of like the definition of hydrophilic. It's small enough, it's neutral that it can go through, but you're not going to get charged things going through, at least not without help. Speaking of help, if we have charged things, if we've got bigger hydrophilic things, we're going to need some help. And that help is typically going to happen through proteins that span the membrane. So those integral membrane proteins. Sometimes all you need is one of those selective pores in the case of our passive transport to let things through. But if we have active transport, well, now we're going to need to use energy, but we'll see how we can use pumps. We can use primary active transport, secondary active transport in order to move molecules against the gradients. And this is then going to allow us to maintain gradients. So just letting things through, providing passages for molecules to get through is great to get things through, but once they get through, then you're losing your gradient. So it's going to make it so that you can't just keep moving things through in that passive manner. And so you need to actually use energy to move molecules to maintain the gradient to drive the transport through passive transport, as well as move molecules in order to just move them where you want them. And so I think one of the easiest ways to see the power of this is to think about primary and secondary active transport. We'll get much more into this later, but primary active transport, here you're actively spending energy at the gate to let things through in a different direction than they would want to go thermodynamically. So this is what I was talking about before, how we were maintaining that gradient of our sodium being high outside of the cell and our potassium being high inside of the cell. We're actually going to see how we're going to be hydrolyzing ATP in order to cause a conformational change in this pump that pumps out 3 sodium ions and pumps into potassium ions, moving them against their electrochemical gradient, maintaining that gradient though, so that we can use that gradient to power the movement of other molecules. In this example, we're pumping out the sodium, we're pumping in potassium into these epithelial cells in our intestines from the blood. And then in your intestines, you're going to have a transporter for glucose. And so the glucose is going to have higher concentrations inside of the cells. And so it's going to need to go against its electrochemical gradient. So we're going to say, okay, sodium, you've got it. You're going to want to come into the cell because we've been pumping out the sodium so that we keep the sodium concentration low in the cells. So come on back in. It's going to be thermodynamically favorable for you to do that. You're going to release some energy, and we could use that energy to drive the co-transport of glucose. And so this is just to show you that we can spend energy in order to maintain a gradient, and we can use gradients in order to drive movement. And again, we'll get more into this. Okay, so we've got these gradients. This leads to potential energy. We can have molecules that we want to move along their gradient and molecules that we want to move against their gradients. And it's going to be easier to move molecules along their gradients because that's going to give us energy versus moving molecules against their gradient, which would take energy. But if we just let all the molecules move along the gradient, then we would have no gradient. So we have passive transport along the gradient and active transport against the gradient. And we'll see that we're going to have multiple types of each of these. We'll start with the passive transport. And so start with the energetically simple run, right? So speaking of simple, the simplest type we can have is just simple diffusion. And so this is where we have membrane permeable molecules. So molecules that are penetrating to the membrane is permeable to them. These can diffuse freely through along their electrochemical gradient. And again, this is going to be our nonpolar molecules, the small ones, and really small polar molecules. So they're just going to go along their chemical gradient or their electrochemical gradient, but they're going to be electrically neutral. So that electric part of the equation will be zero. So they're moving along their chemical gradient, basically. But again, unless you do something to use them inside of the cell, unless you keep bringing more outside of the cell, then you're going to lose that gradient because it's just going to diffuse in and diffuse out, and it will reach that point at which the concentrations are even. So we'll see how we need to kind of keep things uneven. Most molecules, however, are not just going to diffuse through that membrane. Remember, our molecules are typically going to be used to being in these aqueous environments, in these aqueous compartments, and so they're going to need help to get from one compartment to another because these barriers are there to divide those compartments. Facilitated diffusion, here we have molecules that move along their gradients through selective channels. And so it's really important that they're moving along selective channels because if we just had these big holes in the membrane, then the membrane would be kind of pointless. Like, yeah, there would be a long line to get in, but you could still get everything in. So we need these to be selective. And often what we're going to want is that not only are they selective for a molecule, but they're selective for moving a molecule only under certain conditions. This is then going to allow us to maintain the gradient and then use that gradient when we're ready. Sometimes with facilitated diffusion, we're moving a single thing, and sometimes we're moving multiple things. And this is also true for transporters that are going to be active transporters, that we can move one thing or multiple things. If we're just moving a single thing, we call that uniport. But if we're moving multiple things through that same transporter at the same time, we call that cotransport. There are different types. So symport, you're moving substrates in the same direction. So same. And antiport, you're moving them in opposite directions. So anti. So they move one molecule one direction and the other molecule the other direction. So symport and antiport are both examples of cotransport. For passive diffusion, these are just going to be these channels that the molecules can move through, and so we'll see how these can be gated channels, but the molecules are going to be diffusing through those channels based on their electrochemical gradient. The channels are going to make it so that there's a hydrophilic passage from one side of the membrane to another. So the solute is going to go from a watery environment to a lipidy environment and back to a watery environment. This membrane is going to be about 30 angstroms long, so you don't need to know these distances again. But what's important to know is that this molecule is going to be hydrated on one side, so it's going to be surrounded by water. You're going to have to give up those water interactions, make interactions to the channel protein, and then make new interactions to water on the other side of the membrane. And so we can talk in terms of free energy and the energy of activation for diffusion. So it's not an enzyme, but we can still talk about this kind of delta G to this transition state for the diffusion. And so this kind of barrier that you've got to get over in order to go through. And you're able to form interactions with the channel that are then going to lower that activation barrier. Similarly to how we saw that an enzyme could form interactions with the transition state in order to lower the activation barrier for an enzyme catalyzed reaction, we can have similar things with our membrane spanning pores, our transporters for our facilitated diffusion. As I mentioned, we want these to be really specific, and they can have this specificity by making specific interactions with the molecules. Making those specific interactions is what's going to make it so that you're lowering that transition state energy. You're making it easier for the molecule to go through, but you're only going to do that if the molecule kind of fits. And these can be really, really picky. So here's a potassium channel, and potassium is going to be bigger than sodium. But this channel is only going to let through potassium. The way that it's able to do this is because the sodium ion on one side of the membrane, remember, it's going to be surrounded by water molecules, it's going to be hydrated, and the hydrated sodium is going to be too big to go through the channel. But if you strip off those waters, well, now it's going to be too small to make the favorable interactions with the pore. Whereas the potassium, it's going to be very happy. It's going to have these perfect interactions that are going to let it through. And note that here we're talking about one of these pores, and so the potassium is moving along its gradient. And so this is not one of those pumps that was spending the energy. It's letting the things through along their gradient, but it's only letting certain things through, and it's letting things through that wouldn't otherwise be able to go through. If we just had those channels open for potassium, those or some other molecule all the time, we would lose that gradient, remember? And so, yes, we could keep pumping things out, but then we'd just be using that gradient kind of uselessly. So this is just transporting things, but it's not actually powering the movement of something against its gradient or anything like that. You're just kind of diffusing your gradient. Maybe you wanted this inside of the cell, so it's still helpful. It's not just useless, but you're not capturing the energy of that movement in order to do something. And so we typically are going to want to have those channels be gated so we're not just diffusing our gradients all the time. Then we can have them open in response to a stimulus, and this can even cause things like cascading signaling events inside of the cells and various regulatory things if you then open it on demand. So how do we tell the channel to open? This depends. We can have these be ligand gated or they can be voltage gated. And so ligand gated channels are going to open in response to binding of a ligand. Here's an example of a ligand gated ion channel, our nicotinic or acetylcholine receptor. Unlike that potassium channel we were just looking at, this one is permeable to both sodium and potassium. So you'll see there's a lot of redundancy. We have a lot of channels that let through the same thing, or channels that let through the same thing but at different times or in different cell types, various things like that. But this channel is going to let through both sodium and potassium. And what happens is it's only going to do that if acetylcholine is binding to a receptor. And so this is really important for our nervous system. You have acetylcholine that is going to bind to the receptor, cause the receptor to change shape, a conformational change that's going to open up the channel so that the sodium and potassium ions can go through. So you can see that you kind of got this funnel entrance, but it's locked off until you get that conformational change from the binding of the acetylcholine ligand. That'll open the channel so that those ions can get through. You can also have voltage-gated ion channels. So we talked about how inside of the cell was typically more negatively charged, and outside the cell we had a negative membrane potential. In response to nervous system stimuli, so neurons communicating and stuff, they often use electricity. And so you can have that membrane polarization be altered. You can have changes in the voltage, and these changes in the voltage can cause changes in the channel proteins that then cause them to open or close. So if we think about the charged parts of a protein, so we've got the termini where we've got those amino groups, and then we've got the carboxylate groups. Those are going to have charges. We have charged amino acids throughout our proteins. These are going to respond to that change in the charge. You've got the positive going to be attracted to negative and negative to positive and positive repelled by positive and negative repelled by negative. All of those are going to cause the protein to change shape a little to undergo a conformational change that will then open or close the channel. So that would be an example of a voltage-gated ion channel. And these are important for signaling, and having these channels open in response to a stimulus can therefore cause a variety of effects. And so we'll talk about these other types of signaling. So our G protein coupled receptors and our receptor enzymes, like our receptor tyrosine kinases. And then we have gated ion channels. And then we'll also see how we can have steroids that are able to just do that simple diffusion through the membrane, go through the membrane into our nucleus as well, and control transcription, control the making of messenger RNA transcripts from the DNA genes for specific proteins, and that will allow different proteins to be made. In both of these forms, the gated ion channel and the steroids are relying on movement through the membrane. Whereas with our other types of receptors, as we'll see, there's going to be a messenger outside of the cell that's going to cause a change inside of the cell through changes in membrane spanning proteins. And so that's coming up when we talk about the signal transduction, so getting a message from outside of the cell to inside the cell. And we'll talk more about that later. But for now, just no gated ion channels, and then that steroids can freely diffuse through the membrane. Okay, speaking of diffusion and stuff, we're done talking about that simple stuff. We're done talking about the facilitated diffusion. We're done talking about all that passive stuff for now. And now let's go and talk about some active transport. So remember, with active transport, we're going to be needing to use energy in order to move a molecule against its gradient. There are two ways that we can do this, or two basic forms of this: primary active transport and secondary active transport. So primary, this is where we were paying at the door, we're paying at the gate. So we're going to couple the movement to energy producing chemical reactions, typically the hydrolysis of ATP. And we have secondary active transport, where we couple the movement of a molecule against its gradient, so a molecule that requires energy to move. We get that energy by coupling it to the movement of another molecule along its gradient. So a molecule produces energy by moving along its gradient that can be used to drive the movement of a different molecule against its gradient. Primary active transporters are also known as pumps a lot of the time. And often what's going to happen is they have alternating gates. And so you have one gate open on one side that's going to let molecules in, and then you have spend the energy, and then the other gate opens up. There are a few main types: ABC transporters, P-type ACPases, and V and F-type ATPases. I don't need you to know the names of these different types, but you might see these terms used, especially ABC transporters. These are going to be important biomedically, and we'll talk about a little in a second. But these are going to be ATP-dependent transporters that typically are going to use hydrolysis of ATP in order to cause a conformational change to let things through. Whereas P-type ATPases, here, these are going to involve phosphorylation of the transporter as part of the thing. So here you're just kind of hydrolyzing ATP, versus with a P-type ATPases, you're phosphorylating the transporter, and that the P-type ATPases are going to be transporting cations. And then you have V and F-type ATPases that are ATP-dependent transporters that transport protons. And so don't worry about these for now, but we will look at an example of an ABC transporter, as well as an example of a P-type ATPase. ABC transporters, these pump a variety of different substrates, and the reason why I said they were important biomedically is because many of these substrates include things like drugs and amino acids, and also because mutations in them can cause various diseases, and I'll show you in a couple examples in a second. What these have in common is that they have these two ATP binding domains, and so these parts that bind ATP, each of those binds one ATP, so you have two ATP total. And then they have these two transmembrane domains, so these two parts that go through the membrane. In their kind of baseline state, they're facing one side, then they hydrolyze the ATP to give you ADP and orthophosphate that causes a change in the shape. That change in the shape is going to make it so that the other side of the pore is open, and this is going to then allow the molecule, that substrate molecule, to get through. And remember, it's the hydrolysis of that ATP that's providing the energy, not breaking that bond. An example of an ABC transporter that you might come across is CFTR. It's a chlorine channel, and it's really important, and mutations in it cause cystic fibrosis. There are other ones that you can see are involved in cholesterol transport, involved in vision, as well as involved in resistance to various drugs. So lots of ABC transporters in humans, and these are just some of them. We also have a lot of P-type ATPases, and so these are going to transport cations, so our positively charged ions, things like calcium, sodium, potassium. And we were looking at an example before where we had that sodium potassium ATPase. There's also this sarcoplasmic endoplasmic reticulum calcium ATPase, or SERCA pump, and the plasma membrane calcium ATPase pump. And these are uniporters for calcium ions. Again, you don't need to know these names. These are just a couple of examples. What is important about these ATPases is that these P-type ones are going to get phosphorylated. And so think P, phosphorylation, the transporter itself, the pump itself, is going to get phosphorylated. You're not just hydrolyzing the ATP. So there are domains on this transporter that are actually going to get phosphorylated, and that phosphorylation is then going to cause a conformational change that is going to change where things can go through the channel. Binding ATP, but you're not just binding the ATP, you're actually going to be transferring the phosphoryl group from that ATP to the pump. That is then going to change the shape of the pump so that you're going to open to allow the substrate to go to the other side. And typically this phosphorylation is not going to happen until you have that substrate bound in place. So in the example of our circuit pump, so remember this is going to be pumping that calcium and it's uniport, so we're only pumping calcium, but we're actually pumping 2 calcium molecules at a time. So you can see the calcium molecules are bound right here. They've come in from this side, they've bound, we get this shape change, and now we're in this conformation that's going to be ready to go and get phosphorylated. The ATP is going to bind, the phosphoryl group is going to get transferred, so we're phosphorylating the pump. That phosphorylation is then going to cause conformational changes. Those conformational changes are now going to release the calcium to the other side of the transporter. And then this domain is going to reset. And so now you can have another calcium come in. When that calcium comes in, then you're going to get in that conformation to which you can get phosphorylated and then release things. So everything is kind of happening in response initially to the substrate being there, because you don't want to just be doing this unless you're ready to transport. And so that was our primary active transport. We're directly paying at the gate. Now let's talk about secondary active transport, where our friend's paying the cost ahead of time for us. In secondary active transport, we're going to couple the movement of a molecule against its gradient, so an energy requiring process, to the movement of another molecule along its gradient, an energy producing process. But remember that in order to have a molecule have a gradient, we then needed to be spending energy in order to maintain that gradient. And that's where the energy spending comes into play, is to create the gradient that then is used to power the movement of another molecule. An example of this is our sodium glucose symporter. So symporter, it's transporting sodium and glucose in the same direction. It takes up glucose from the intestine in a process driven by the downhill flow of sodium ions. So remember that we keep sodium concentrations low inside of cells, but we do that by pumping sodium out of cells actively through that sodium-potassium ATPase. So we have the sodium come into the cells passively. It wants to get in because of the electrochemical gradient. It's going to need help because it's charged, but we can use facilitated diffusion to get it in. When we get it in, however, we're going to pump it out so that we can keep the concentrations low in here in order to maintain this gradient. Because if we didn't keep pumping it out, then it would build up inside the cells, and then we'd reach equilibrium. We'd have equal concentrations inside and outside of the cells. That would not be good. We'd lose our gradient. And we need that gradient in order to do things like, in this case, move glucose. Glucose is going to be higher inside of our cells, and so we're going to need to not only give it a channel to go through, but also give it some energy to move. In order to do this, we are going to use the gradient of the sodium, transport sodium and glucose through the channel at the same time in the same direction. So we've got symport, so a cotransport because we're moving 2 different things, and symport because we're moving the same direction. This one, though, is going along its gradient, whereas glucose is going against its gradient. Inside of our intestinal cells, now we have this high concentration of glucose. That's why we had to use energy to bring glucose in. But on the other side, in our blood, here we actually have a low concentration of glucose. So we can use a glucose uniporter that is just going to use facilitated diffusion to transport that glucose molecule, to just give it a channel to go through in order to get into the blood along its gradient. But we had to spend energy in order to get it into the cells. And then we're able to maintain that gradient, but then we can use that gradient to get it into the blood. So the primary active transport of the sodium is going to power the gradient that is then going to be used to co-import glucose in secondary active transport. So to review, transport can be passive along a gradient or active against the gradient. When we talk about gradients, we can talk about chemical gradients, electrical gradients, and the combination of the two, an electrochemical gradient. These gradients lead to potential energy. Molecules want to move along the gradient and don't want to move against the gradient. But we can make them move against the gradient if we provide energy. And if we don't keep things from just moving along the gradient, then we will lose the gradient. Even if things want to go through, they can't, often without help. And this help is typically going to come from integral membrane proteins, or IMPs. These go through the membrane, often through alpha helices and beta strands and beta barrels, and they allow for channels through the membrane. Sometimes all you need is a channel, and then you can do facilitated diffusion if you're moving along a gradient. You just need a channel that kind of matches your shape and matches your charge and has all those favorable interactions that are going to make it a nice journey for you. But that channel might be gated, so it's only going to open up in response to a stimulus, like a ligand binding or like a change in voltage. Some molecules, however, are going to need to spend some energy to get them through. We need active transport, we're moving against the gradient. We can have primary active transport, where we're paying at the pump, or secondary active transport, where someone's paying ahead for us. So passive transport, the passage for you is free because you're thermodynamically wanted on the other side. You're moving along your electrochemical gradient. With simple diffusion, you just sneak in, and that's only possible for small polar molecules and smallish nonpolar molecules. Facilitated diffusion, here a bodyguard, so some sort of membrane-spanning protein is going to help you get through. We have selective pores that have channel structures that only permit the movement of specific molecules. These are typically gated, so they only open in response to a stimulus, such as binding of a specific molecule in the case of a ligand-gated channel, or changes in voltage across the membrane in the case of voltage-gated channels. Molecules that are moving against their gradients, that are not thermodynamically wanted on the other side, are going to need active transport. So the passage needs to be paid for energetically. With primary active transport, you pay for your own entry or exit at the gate. And this typically involves pumps that spend ATP to change the shape of the pump via phosphorylation and/or ATP hydrolysis, so that burning of ATP. You can also have secondary active transport, where your friend pays ahead of you. You use primary active transport of one molecule to create an electrochemical gradient of that molecule. Subsequent movement of that molecule back down its gradient is then coupled to the co-transport of a second molecule against its gradient. Voila, that is how molecules move, or don't move, or at least move controllably through membranes.